So an important topic we have to discuss about is neuromuscular junction and the events taking place at it. So before we go into how the nerve communicates with the muscle that causes voluntary movement, meaning your skeletal muscle, we'll just talk about how movement originates from the higher centers in your brain, voluntary movement. So when it comes to voluntary movement, it's what you want to take place. Voluntary movement, like you intend to pick up a mobile phone in your hand, or you want to pick up a pencil from a desk, or open a door or something like that. So for that, you make the intention. So the intention or the idea begins in the front of your brain, in this area called the prefrontal cortex and other association areas of the cortex that give this idea. So what happens is, from here, now the idea has to manifest into reality. So you could see this green arrow that's pointing over here, and this is where the order goes to the motor cortex, the intention, the idea of having to pick up a pen or whatever the intention is. So now the motor cortex has to get a plan, and so for that it goes and gives signals to the basal ganglia and cerebellum, asking for a good plan, exactly how much force, which muscle, what to activate in what sequence. So the cerebellum and the basal ganglia give the idea, give the blueprints of the plan back to the motor cortex, and finally the motor cortex gives rise to descending tracts that go down the spinal cord, and eventually there will be this one particular neuron that is about to come out of the spinal cord, and this is your alpha motor neuron. What do we call this? Alpha motor neuron, which is going to synapse with your muscle over here, the muscle. So here you have the alpha motor neuron which synapses, and we are going to look at this small area over here. Okay, so just remember, intention is created in the prefrontal cortex, and from this prefrontal area and the association areas of the cortex, it will go to the motor cortex. It will, with the idea, I want you to pick up the pen. So the body must pick up the pen and pick it up in a maybe fast or slow way depending on the situation. Then after that, the motor cortex says, I need a plan, so I'm going to go to the basal ganglia and to the cerebellum for advice. Which muscle should I activate first, then which muscle or which group of muscles, and how much force, how much intensity. I need a proper plan, and in which sequence, like that. So basal ganglia and cerebellum use the right coordination, control, force, initiation of movement. All of that takes place. Signals go back to the motor cortex, and finally down the spinal cord to the alpha motor neuron, which will go to the skeletal muscle, which we are talking about over here. Skeletal muscle. Okay, so let's get into the proper topic over here. Number one is structure. First we need to see about structure of the NMJ, neuromuscular junction. Okay, so now let's see. If we look at the alpha motor neuron that is coming out of the spinal cord, here is your neuron. So the various dendrites are over there. And now what happens is, we are into the structure of the neuromuscular junction. Okay, so here we have a neuron, an alpha motor neuron. If we remember alpha motor neuron from Erlanger-Gassier classification, we have got this neuron which is quite long, depending upon how far your muscle is from the spinal cord, because this neuron is in your spinal cord over there. Okay, so it has to go all the way, sometimes from your back all the way to the little muscles in your feet. So it can be long, it can sometimes be short also. So these neurons are quite long, they're myelinated, they've got myelin that allows for faster conduction, also known as saltatory conduction, to take place, because it is more energy efficient and it transmits the signal quicker. So here we have the nodes of Ranvier with voltage-gated sodium channels, same ones we spoke about in your action potential. So each of these nodes of Ranvier will have a few sodium channels that will transmit the depolarization, the action potential, quickly jumping. It jumps from one node of one way to the next, the signal. But when we reach the end of this neuron, what happens is the shape is different. This axon, it will branch. This alpha motor neuron will make branches over there. This is called terminal arborization. Terminal meaning at the end, arborization. Remember, arboreal meaning branching, like arboreal animals like the monkey, because they climb from branch to branch. So that's what arborization refers to, branching. Okay, so it's a fancy word for making different branches. Why? Because one alpha motor neuron will innervate many different muscle cells in the same muscle. Right, that's one more thing you should be aware of. It's not always going to be that one alpha motor neuron will only innervate one muscle. That can happen. Sometimes there can be more than one muscle, okay, depending upon the muscle fiber. If you want very accurate movements, you get these arborizations, which are very restricted in number. So you have very one neuron innervating only a few muscle fibers. So this allows for very fine tuning of movements, so very accurate movements, like in your fingers, where you use for very fine work, like knitting, stitching, or even typing on a small iPhone. So you need very accurate movements of your thumb and of your finger. So those kinds of muscles are innervated by alpha motor neurons, which branch and supply only a few muscle fibers at the same time. Anyway, let's go into one of these areas where the synapse is taking place. So the synapse is the meeting point with the neuron and maybe another tissue, meaning a target tissue. It could be another neuron, it could be a muscle, it could be various different tissues over there. Okay, so now we'll talk about this one particular one, which is pertaining to the neuromuscular junction. So this is the axon, this is the myelin over here. So this is how I would draw the structure. This is the myelin with the Schwann cells over here. You can write Schwann cells. There's the thing that goes over there. This is some voltage-gated sodium channels, because you want to show that the nodes of Ranvier have voltage-gated sodium channels. Okay, and Schwann cells have little nuclei also over here, so we can draw that there. And then we are just looking at one of these branches, and this is your terminal bouton. Bouton is like a button. So this is a button. That's the thread that's holding the button to your coat. Think of it that way. So terminal bouton. And if we look at the structure of the terminal bouton, we'll just talk about that in a little more detail. So I'll just maybe expand this diagram a little more. Yeah, there, that's better. So here we have a terminal bouton, and here this terminal bouton has a lot of vesicles. These vesicles are called synaptic vesicles, of course. Synaptic vesicles, also known as quanta, because they have a quantity of acetylcholine molecules in them. How many? 10,000 each. 10,000 in each of these. Okay, so I've drawn a few. And this area also has a lot of mitochondria that help in making the acetylcholine. So I'll just draw this as a mitochondria. So you draw a little mitochondria over there. Okay, and then what else do you have over here? Well, you have this area called active zones, where these vesicles containing acetylcholine will fuse when an action potential comes down. So there are some areas called active zones, and they are located next to these bar-like structures called longitudinal dense bars. So they are here, these dark structures that I'm drawing here. They extend longitudinally, but because we've made a section of your nerve, we are not seeing that over here. I've not depicted that, but they extend along the length over here, but just ignore that for now. Just to know that next to these longitudinal dense bars are the active zones, where certain structures are existing over here. These structures, let's draw them in a different color. Let's make it yeah, red, because it's nice and active. There we go, red over here. These little triangles I'm drawing are some very important structures called T-snare. T-snare, okay, target snare. It's target snare protein, okay. So this T-snare is like, it fits like a glove in one of these molecules that coats your vesicles. So your vesicles have these molecules that fit perfectly with the T-snare, and we call these V-snare. What do we call these? V-snare, vesicular snare protein. V-snare, T-snare protein. So what happens is, action potential comes down this axon, and then it's going to cause sodium to come inside. So sodium enters in, spreads and opens more sodium channels, so more positivity spreads. You're causing depolarization and action potential to move down until we reach the terminal boson. Now I forgot to mention there's an important ion channel located in the terminal boson. This is called your voltage gated calcium channels. Voltage gated calcium channels. So here calcium can enter into the cell. So you get the action potential flowing down the nodes of Ranvier, and then you reach the terminal boson. And then here, instead of sodium entering in, you get calcium entering in. Now why? Because when calcium enters into a cell, usually something has to leave that cell. That's how it works. If a neuron has to release neurotransmitter, calcium comes into that synaptic area, allowing the release of neurotransmitter. If your hormone secreting cells have to release a hormone, calcium has to enter that cell. Calcium plays a very important role. So for example, if you want to secrete insulin, sugar levels rise, but eventually the sugar levels will enter the beta cells, and it will cause the beta cells to cause some voltage changes. We'll talk about in the endocrine chapter, and it will open voltage gated calcium channels there, and that will cause the release of insulin. So if you want to release something from a cell, especially if it's secretory or a neuron cell with a chemical synapse, then you need calcium to enter. There are only two exceptions to this: parathyroid hormone and renin, from the renin-angiotensin-aldosterone system. Just be aware of this. This is good even for your entrance exam, competitive exam, okay? So now if we look at this, the calcium enters in because of the depolarization, the wave of depolarization that has spread across this membrane, and it is going to cause a few of these vesicles, around 100 to 150, to fuse with the T-snare. So the V-snare and the T-snare fit perfectly like a little glove. It fits beautifully over there, okay? When they fuse together, then exocytosis takes place of the acetylcholine molecules through the vesicles or through the quanta. Right? So the calcium causes the V-snares to go searching for the T-snares, and then they fuse together and release some neurotransmitter into this space. Now, what is this? What I'm drawing here now is the surface of the muscle cell, the cell membrane of the muscle. Now the cell membrane of the muscle is like a trough, okay? It's like a trough. You know what a trough is, right? You know, you have some horses or cows and you have to feed them water. So, you know, there's a tub that you fill with water. So that's called a trough. So this is like a tub for the acetylcholine molecules to, the neurotransmitter molecules to, you know, to land into, or there to fill up, so to say. So here we have a space existing between your neuron and the muscle fiber, and this is your synaptic cleft. This is your cleft, synaptic cleft. Okay, so synaptic cleft. I'll just move that over there. You just remember this as the T-snare over here. T-snare, T-snare. I just need a little more space over here. So now, this is your synaptic cleft. It's around 100 nanometers in the middle over here. So this is around 100 nanometers from this distance. And actually, don't let this diagram confuse you. Actually, the synaptic cleft, if we see, it's more like this. 100 nanometers distance in the middle, only a few 10 nanometers in the middle over there, up to 1 nanometer almost to the edge over here. So just be aware of this also. Okay, so now we have this cleft across which the neurotransmitter chemical will be released. Now look at this. This is the postsynaptic membrane. This structure of the skeletal muscle is the postsynaptic membrane. Here we have got your receptors. These are ligand-gated channels. What does that mean? That means a chemical sticks to this, some ligand or chemical sticks to it, and it will cause the opening of these channels. There's a gate over here. There's a gate inside this. And if the molecule, two molecules actually of acetylcholine molecules bind over here and find this place and bind over here, it will open the gate. Then what will happen? Cations, especially sodium, will come in, causing depolarization and making the cell depolarized over here. So this is how it works. Sodium will depolarize over here. Now look at this. We have the sodium entering, depolarization taking place over here, and this will cause your action potential to form over here. So this area where you could see the ups and the downs, these remind you of wave patterns. So these are like crests and troughs. Do you remember crests and troughs from the study of waves in physics over there? So there we go, crests and troughs over there. Okay, so on top of the crests are concentrated these ligand-gated channels, and these are called nicotinic. They're also called nicotinic M type. Nicotinic, you can also say nicotinic M type cholinergic, because they are activated by both nicotine and acetylcholine. So if you are exposed to nicotine, the nicotine molecules can do the same work as acetylcholine does over here. Now don't take up smoking, that's bad for you. Though it might increase your reaction time because there will be some nicotine floating in your system, but that is not an excuse to smoke. It's always a poison. Okay, so be healthy. Nicotinic M type of cholinergic, this one, channels, ligand-gated channels, ligand-gated channels. So we don't need this in green over here right now. Nicotinic M, M1 you can also say, or no, just say M. M is good enough. Nicotinic M type of channels. Okay, M for muscle. That's how you remember this. Cholinergic ligand-gated channels. So they will bind over here. If you release enough acetylcholine, then you will have enough sodium causing enough depolarization that you will create a chain reaction called action potential. Please watch the action potential video to understand how this works, and that will then spread down along the membrane of the muscle until it reaches something called the T-tubules, which will give a signal to what we call the L-tubules, which release calcium in the cytoplasm of the muscle, causing excitation, contraction, coupling. Excitation, contraction, coupling. So you need to understand the structure of the NMJ, neuromuscular junction, before we can talk about the sequence. Now I've already told you the sequence, but I'm going to again tell you about the sequence in a more organized and algorithmic fashion in just a minute.